Hello friends, welcome. Let's discuss mitosis in this video today. What is mitosis and what actually happens in it? Mitosis is nothing but a phenomena in which one cell gets divided into two cells, and those two cells again gets divided into four. In this way, the entire body is generated out of one cell. So let's see how this procedure takes place in our body. Mitosis is divided into two phases: interphase and mitotic phase. Interphase is a phase where a cell spends much of its time, and it takes less time for a cell to spend in mitotic phase. Now, interphase is divided into G1, S and G2 phase, whereas mitotic phase is divided into prophase, metaphase, anaphase, telophase and cytokinesis. G1 phase is a phase where cell growth happens, S phase where DNA synthesis takes place, and G2 phase where again cell growth continues, and then the cell enters the mitotic phase. Let's see the cell cycle in this. This is a cell cycle where a cell will be in G1 phase for cell growth. The cellular organelles get synthesized, proteins and RNA synthesis takes place in G1 phase. After that, cell enters the S phase, also known as synthesis phase, in which the DNA is replicated. It is duplicated to be divided into both the cells. And then the cell enters G2 phase, in which the cell growth again continues, and cell prepares itself to enter the mitotic phase. The M phase here is a mitotic phase. Now let's see the mitotic phase. What actually happens? In mitotic phase, when the cell is in interphase, the chromosomes look like thread-like structure, and there will be two pairs of centrioles. When the cell enters the prophase, the two centrioles, one pair here and one pair here, the two centrioles move towards the opposite poles of nucleus. Why do they do that? To form a spindle, over here. Centrioles will prepare spindle fibers. Centrioles will form spindle fibers. That's why the centrioles move towards the opposite poles of nucleus. Now, in the prophase, the chromosomes which were looking like thread-like structure over here, they will look like the stainable, thick and condensed chromosomes, like this. Condensed means to get shortened. Now the cell enters metaphase. In the metaphase, the chromosomes are aligned in the middle. Metaphase means they are aligned on the metaphase plate. Why they are aligned? We can understand that in anaphase. They are aligned so that they can be pulled apart. In anaphase, the chromatids are pulled apart from each other. And in telophase, they get separated. The nuclear envelope is again formed and they get separated. Whereas in cytokinesis, what happens is we get two individual cells. This is how one cell gets divided into two cells. I hope the video was helpful to you. Thank you so much.